Hello everyone, welcome back to the channel. In previous video we have studied outline of fungal classification as per the system proposed by Alexopoulos and Mims. Today we will focus on division Gymnomycota, also known as slime molds. Now by the end of this video you will clearly understand every key point in a super simple way. Fungi are placed in kingdom called Mycetae and they have some unique characteristics. Let's begin with the basics. All fungi, including slime molds, are placed in a kingdom called Mycetae, which is further divided into these three divisions: Gymnomycota, Mastigomycota, and Amastigomycota. Now, what makes these fungi special? Let's understand each characteristic one by one. First is kingdom Mycetae. Here the members are achlorophyllous. That means fungi do not contain chlorophyll. This means they cannot perform photosynthesis like plants. So they depend on other sources for food. The members of kingdom Mycetae are saprophytic or parasitic. That means fungi can obtain food in two ways. First, they can live as saprophytes. That means they feed on dead, decaying matter. Or they can live as parasites. That means they feed on living organisms. Next is about their body structure, that is thallus. Thallus is either unicellular or it is multicellular. For unicellular thallus, you can take an example of yeast, and for multicellular fungi, you can take the examples of molds like rhizopus and mushrooms like agaricus. Next is about their cell wall. Cell wall consists of mainly chitin. That means, unlike plants, fungi have a cell wall made of chitin. But in some members, you will find that their cell wall is made up of chitin as well as cellulose. So in some members, you will find small amount of cellulose in it. And hence, the point mentioned here is what? Cell wall consists of mainly chitin. Next is about mode of nutrition. It can be either absorptive or phagotrophic. So most fungi absorb nutrients from their surroundings, but some use phagocytosis, meaning they engulf food particles. Next is about asexual reproduction. It occurs by formation of spores which can spread easily through air and water. Most fungi also reproduce sexually. That means sexual reproduction is usually present. Now let's study first division, that is division Gymnomycota. Gymnomycota includes slime molds which are quite different from typical fungi. Now let's study what are their key features, what are their characteristics. Here the members are phagotrophic, that means they engulf food particles instead of absorbing them, and their somatic structures are devoid of cell wall. That means their body lacks a cell wall in the vegetative stage. So here only the spores are surrounded by cell wall. Rest of other vegetative structures, they are devoid of cell wall. Now the characteristics of division Mastigomycota and Amastigomycota we will be discussing in upcoming videos. Now let's focus only on division Gymnomycota. Division Gymnomycota is further divided into two subdivisions, that is Acrasiogymnomycotina and Plasmodiogymnomycotina. First is subdivision 1, that is Acrasiogymnomycotina. Here, here the organism starts as mixed amoeba. That means amoeba-like cell. So here soma or cell is mixed amoeba, followed by pseudoplasmodium that terminates into a sporocarp. So here the organism starts as mixed amoeba, that is amoeba-like cells. These cells come together to form a pseudoplasmodium, that is a temporary aggregation. This, this develops into a sporocarp which produces spores, and spores have cell wall. So here the key point is what? Cells, that is mixed amoebae, they stay separate but cooperate. It means the plasmodium is what? It is pseudoplasmodium. Next is subdivision 2, Plasmodiogymnomycotina. So these members are called true slime molds. Members of Acrasiogymnomycotina, they are called cellular slime molds. Okay. Subdivision 2, Plasmodiogymnomycotina. What is the first characteristic given? Soma is mixed amoeba followed by true plasmodium terminating into one or more sporophores. Spores are produced at the tip of the sporophores and are with cell wall. That means here the members, they start as mixed amoeba but then they form true plasmodium. That means a large multinucleate mass. The plasmodium develops sporophores that produce spores, and again the spores have what? Cell wall. Now next is class of Acrasiogymnomycotina. Subdivision Acrasiogymnomycotina has single class Acrasiomycetes. In case of Acrasiomycetes, you will know flagellated cells are produced, but there is one exception. So, except one species, mixed amoebae aggregate to produce pseudoplasmodium. Pseudoplasmodia are lobose, that means blunt or broad, or they are phyllos, that means they are thin or thread-like. Sexual reproduction occurs through microcyst. Now, class Acrasiomycetes is divided into two classes, Acrasiomycetidae and Dictyosteliomycetidae. Class 1 Acrasiomycetidae, what are the characteristics given? Aggregating mixed amoebae does not form streams in developing pseudoplasmodia. Sorocarps with or without definite sori or sporophores. Mixed amoebae with lobose pseudopodia and nuclei with single centrally located nucleoli. Subclass Acrasiomycetidae has only one order Acrasiales, and example is Acrasis. Next is subclass 2, that is Dictyosteliomycetidae. Here, aggregating mixed amoebae, they form convergent streams. Sporocarps are with well-defined sori and sporophores. Mixed amoebae with phylloid pseudopodia and nuclei with two or more, so two or many peripherally located nucleoli. So these are the distinguishing points for Acrasiomycetidae and Dictyosteliomycetidae. Subclass Dictyosteliomycetidae has only one order Dictyosteliales, and example is Dictyostelia. Next is subdivision 2, Plasmodiogymnomycotina. It is further divided into two classes, protosteliomycetes and myxomycetes. Now let's study what are the characteristics of class 1 protosteliomycetes. Here, mixed amoebae produce sporocarp directly or after the development of plasmodia. Plasmodial streaming is unidirectional. That means the movement is unidirectional and they produce flagellated cells, generally biflagellated cells. Class protosteliomycetes has only one order, protosteliales. Example is protostelium. Next is class 2, that is myxomycetes. Here, mixed amoebae fuse to form zygote which develops into plasmodia on which you will get various types of sporophores. Plasmodial streaming is reversible. So here movement is reversible. Meiosis occurs in pre-spore condition or in spores. Now this myxomycetes class is further divided into three subclasses: Serratiomyxomycetidae, Myxogastromycetidae and Stimonetomycetidae. Now let's study their characteristics. Subclass 1, Serratiomyxomycetidae. Spores are borne externally, singly at the tips of the hair-like stalks. Sporophores are columnar, dendroid or poroid. Subclass 2, Myxogastromycetidae. In these members, spores are borne within the peridium and in masses on various types of sporophores. Sporophore development is myxogastroid. Next is subclass 3, Stimonetomycetidae. Here, spores are borne within peridium and in masses on various types of sporophores. But here, the development of sporophore is stimonetoid. Now for different, okay. Subclass Serratiomyxomycetidae has only one order, Serratiomyxils. Example is Serratiomyxa. Subclass 2, Myxogastromycetidae, has four orders, namely lycials, echinostelials, trichials and physaries. Now let's study their characteristics. In case of lycials, you will get true capricium and columella is absent. Spores are brightly colored and example is Cribraria. Order 2 is echinostelials. Example is Echinostelium. True capricium is present. Sporophore is minute stalked. Columella present. Spores brightly colored. Next is order 3, trichials. Example is Calomyxa. True capricium is present. Sporophore large stalked or sessile. Columella is absent. Spores are brightly colored. Order 4 is Physaries. Example is Physarum. True capricium is present. Spores are black or deep violet in color. Now next is subclass 3, that is Stimonetomycetidae. It has single order Stimonites. Example is Stimonitis. Here you will get capricial network or thread on surface of sporangium. Columella is present and spores are violet brown in color. So this was about division Gymnomycota. In upcoming video, we are going to study the characteristics of division Mastigomycota. If